The overall goal of this simple in vitro co-culture model is to mimic the characteristics of an in vivo tumor microenvironment, enrich individual cell populations, and investigate various modes of intercellular communication.
This method can help answer key questions in the cancer and radiobiology fields, specifically regarding the factors underlying the generation of cancer-associated fibroblasts and the responses to therapeutic agents.
The main advantage of this technique is that it allows the generation of individual cell populations from a mixed cell culture without stress-inducing fluorescent tagging or cell sorting.
Begin by washing the cell culture of interest 2 times with 5 milliliters of PBS. After the second wash, detach the cells with 1 milliliter of room temperature trypsin EDTA for 2 minutes at room temperature. Then quench the reaction with 9 milliliters of complete growth medium, gently pipetting the medium over the surface of the flask 10 times to dissociate the cells. After counting, dilute the cells to a 2.5 times 10 to the 5th cells per milliliter concentration in fresh medium, and transfer the cells to a sterile 15 milliliter centrifuge tube. Spin down the cells by centrifugation, and resuspend the pellet in fresh growth medium supplemented with 50% fetal bovine serum at a 2.5 times 10 to the 5th cells per 70 microliters of medium concentration. Next, transfer inserts of the appropriate experimental pore size from their packaging into individual wells of a multi-well dish and cover the dish.